So now today I'm starting my second chapter, that is sexual reproduction in flowering plants. Here you can see the diagram of a flower in which here it is stigma, anther, petal, filament, style and sepal. And this is ovary. The organ specialized to perform sexual reproduction in angiosperms are flowers. Flowers are basically modified condensed shoots. A typical flower has 4 rolls of flower leaves, sepals, calyx, petals, corolla, stamens, androecium male reproductive part, carpels, gynoecium female reproductive part. The male reproductive organ, stamen. It is the male reproductive unit of an angiospermous flower. Okay. It consists of 2 parts. A long and a slender stalk called filament and a bilobe structure called anther. It's anther and it's a filament. Proximal end of a filament is attached to the thalamus. This is the end which is attached to the thallus. The anther is usually bilobed with each lobe having 2 theca, that is, they are dithecous. 2 theca means dithecous. The anther is 4-sided. 4-sided means tetragonal. It consists of 4 microsporangia located at the corners, 2 in each lobe. Now here we go with the structure of a microsporangium. It appears circular in outline and surrounded by 4 wall layers. So here you can see microsporangia develop into pollen sacs and they extend longitudinally. So this is connective and this is epidermis layer, endothecium, sporogenesis and middle layer. Epidermis, endothecium, their function is to protect, and middle layer, their function is to protection and the dehiscence and look before the dehiscence of anther. And tapetum provides nourishment to the pollen grain. First we see epidermis, one thick and a protective in function. Endothecium, single layer. Cells have cellulose thickening wall with a little pectin and lignin. In some eases, it helps in anther dehiscence, splitting at maturity. Middle layer, 1 to 6 layers. They degenerate during maturity of the anther. Tapetum. Tapetal cells are nutritive. By this, rubbish bodies are produced with help in ornamentation of microspore wall. A compound sporopollenin is secreted by which is deposited in the exine of the microspore wall. The cells of tapetum possesses dense cytoplasm and generally have more than 1 nucleus. When the anther is young, a group of compactly arranged homogeneous cells are called sporogenous tissue occupies the center of each microsporangium. Okay, so next topic is our microsporogenesis. These notes contain all the main points of NCERT and some important things that you should know for giving your competitive examinations. Microsporogenesis. Anthers develop, cells of sporogenous tissue will undergo meiotic division to form microspore tetrads. As each cell is capable of giving rise to microspore tetrad, each one is potential pollen or microspore mother cell. This is known as PMC, pollen mother cell. The formation of microspore from a pollen mother cell through meiosis is called microsporogenesis. The formation of microspore from pollen mother cell is called through meiosis is called microsporogenesis. Kindly see that you don't miss any word. If you miss any word, then the meaning of whole sentence will be changed. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed. They are released with the dehiscence of anther. So here we complete our first video, and in next video we are going to read about pollen grain. Kindly like, share, comment and subscribe my channel.